Hi everyone, my name is Bharavi, and I would like to talk about my study on unraveling the genetic basis of prostate cancer phenotype in India. My supervisors are Dr. Prashant Sudvajala, PI of System Genomics Lab at Amrita School of Biotech and the founder of Bioclues, and Dr. Rajguru Aradhya, Assistant Professor at Developmental Biology Lab at Amrita School of Biotech. The objectives of my study are as follows: to explore the genetic variations associated with prostate cancer phenotype in India through whole exome sequencing, and perform comparison of consensus variants from our dataset to the data from Practical Consortium to identify commonalities between Indian and European phenotype of prostate cancer. The rationale of our study are as follows: prostate cancer burden in India is rising. It is the third most prevalent cancer in India, and the treatment of advanced prostate cancer, especially castration-resistant prostate cancer, is a challenge. This is worsened by the fact that there is an inherent stigma among Indian men, and they refuse to take prostate exams. Hence, cases are reported later, and oftentimes these CRPC cases are lethal upon metastasis. The biological mechanisms underlying the development of prostate cancer are also complex, involving both epigenetic and genetic factors. And Unlike some cancers, prostate cancer has a high heritability of 58%. In order to understand this, we are attempting to perform whole exome sequencing to understand the genetic landscape of prostate cancer in India. The methodology employed in our study are as follows: 39 malignant prostate cancer samples with high Gleason grade between 7 and 9 were collected from Rukmini Birla hospitals in Jaipur, and institutional ethical committee clearance was obtained from Rukmini Birla, and informed consent was taken from all patients. Whole exome sequencing was performed, and the raw reads were analyzed through our in-house benchmark pipeline, namely the Convex pipeline. Gene annotation was performed using ANOVAR, and common variants were compared to the OncoArray chip project from Practical Consortium to obtain common SNPs between Indian and European population. Uh, Here is an outline of the convex pipeline. To summarize this, we are using four different variant callers: BCF tools, VT, VARSCAN, and FreeBIOS, and we are obtaining consensus variants from all four. After which, we are filtering them based on pathogenicity, mineral frequency, and zygosity. The results of our study are depicted here. Um, From 39 samples, 2,561 consensus variants were obtained, out of which 1,669 were exonic, 754 were non-synonymous variants, and among these, key cancer-related genes have been identified: CYP11B2, COL6A1, HEXB, all of which are involved in tumor invasiveness, angiogenesis, and metastasis. We also performed a comparison of our consensus variants to the list of deleterious genes reported by Ravindran et al. This comparison reveals 6 common genes, which are listed here, out of which MUC16 and ERB31 are notable. ERB31 is an endogenous retroviral protein which could possibly serve as a prognostic biomarker for prostate cancer. We also performed a comparison of our consensus variants to the OncoArray data from Practical Consortium. 118 SNPs out of 600,000 that they genotyped were common between the two datasets, representing commonalities between the Indian and the European phenotype of prostate cancer. The whole exome sequencing performed in our study reveals that the genetic landscape of Indian prostate cancer phenotype is diverse, and we are attempting to perform 200 exomes to understand this and maybe identify potential diagnostic or prognostic markers that could serve to better understand and diagnose disease. Thank you.